In the last video, we talked about what transcription is, and we talked about how transcription is different in terms of prokaryotes and eukaryotes, but we haven't really talked about eukaryotic transcription. And that's because eukaryotic transcription and prokaryotic transcription are essentially very similar, and there's only one major difference, and that's the entire processing of mRNA. And we talked about how mRNA is the primary mRNA inside prokaryotes are ready to be translated. However, that's not true with eukaryotes, and the reason we're going to understand just now. So notice how DNA inside eukaryotes are very long, and they have very long sequences. And there's a problem. The problem is that we have areas that are non-coding regions, for example, promoter regions, enhancer sequences, non-target instructions on this DNA that is transcribed. And because we don't need these instructions, they're intervening in the whole process. These regions are intervening. And notice how intervening is written in an awkward way. And that's because I want you to memorize that they are called introns. Introns are intervening sequences, and they have to be expelled. On the other hand, we have genes that are being expressed, and whatever is being expressed is called exons. Exons will be joined together later on. So before mRNA is translated, introns are removed and exons are joined together. And this is the major step from hnRNA to go to mature mRNA that can be translated and sent off to the cytoplasm. This is made possible through splicing, and splicing is mediated by spliceosomes. The spliceosome is a very complex structure. It's a very complex. It's a very complex structure, and this complex is formed with over 100 proteins and small nuclear RNAs. And the thing you want to really quickly understand is that spliceosomes are not like RNA polymers, which are ready to just bind on, just translate and just get out of there. They have to be made on spot because they're so complicated and they contain so many different things. And about half the proteins inside the spliceosomes will bind to the sRNAs, the small nuclear RNAs, and these will form three snRNPs. And remember that the P here stands for a particle, so small nuclear ribosomal nucleic particle. Now these three snRNPs are therefore made of proteins, and they have an important role in the series that will follow. So the complex will undergo many changes, and these changes are made because just to promote catalytic activity. So here's what the entire structure will look like. In a sense, we have these spliceosomes come together, and then we have splicing happening. The orange is the exons, and the red is the introns. We're trying to get rid of that. They're intervening. So the splicing of the reaction really is the snRNPs will recognize an hydrogen bond to the nucleotides on the introns, and this is typically recognized on the 5' end by the GU sequences and the 3' end by the AG sequences. And these sequences will help the recognizing introns for the spliceosomes, and specifically in the spliceosomes, you have the snRNPs who are doing this. So the next thing we want to talk about are the 15 to 45. We also have the 15 to 45 adenine bases that are upstream of the 3' splice site, and these are also regions that are helping recognize the introns. So the introns are the entire process of introns. The spliceosome function happens in a two-step catalytic reaction. The first is the snRNA, the small nuclear RNA, will bind to the 5' and the 3' ends of the introns, so they're going to bind onto it. And the reserved adenine base pair bond. These are the base pair bonds of the adenine. This is a more complex structure that is formed on. And then eventually we have splicing happening, and splicing is when we have the entire spliceosome that's coming off. This is the intron that we don't need, and this is the exons. The mRNA is now ready, and they're joined together. And this is when splicing is completed. And remember that the GU and the AG and the adenines are the hinting. It just highlights, that these are the bases that help recognize the spliceosome. This is the intron, and this needs to be removed. This is intervening. So now the next video we're going to talk about the alternative splicing and the different ways splicing happens.